Good afternoon, Dr. Bala.
Good afternoon, Tarna.
You're able to hear me, sir?
Yeah, it's correct. It's correct. Now I spoke to Professor Pichavan. He is connecting directly.
Okay. So
Yeah,
he's not sending his slides to us?
it seems that he is connecting direct.
Okay.
He's at it. He's at it in a couple of minutes. He will finish the process. It seems that there is a problem in sending the slides. It seems he tried a couple of times, but he was
Okay.
not able to successfully send it.
Is there someone who can help him?
He is good at the computer and IT things. I hope he may be having someone in his office.
Okay.
Yeah, he himself can able to operate nicely, but people are there. People are there in his office.
Okay. Then there should not have been a problem in sending the slides. I don't know what's. Okay, no problem. It's okay. Let's hope for the best.
Yeah. Let us hope for the best.
So you have any information from Professor Rao? Will he join for a while?
Uh, in the morning when he spoke to me, he said that he may not be able to join.
Okay.
Uh,
In response to my message, he said, because
hmm,
I requested him, sir, please join for a short
okay.
while in the beginning so
Oh,
that we can start the session. And he said, okay. So can
okay.
you please give him a call and find out? Yeah.
Yeah. Yes, Dr. Dharma, he said he's joining. I will be joining for a while and
Yes.
not able to speak, it seems, so he will be joining.
Sure. Thank you.
Yeah.
Great. Hi, Ravi.
Hi, Taruna.
You're able to, yeah, we can see you. A little issue with your visibility. A little hazy screen. Dr. Bala, you also find that, or it is me only? So Dr. Bala, you need to start the session, right? Hello.
Yeah. As per
Yeah.
your, yeah, I saw
Yeah.
your mail, and
Thank
accordingly
you.
I will start the session. Yeah.
Thank you. Thank you, sir.
Yeah.
Thank you. Thank you.
I'll invite Ravi to proceed further. Yeah, sure.
Yes. Thank you. Thank you, sir.
Yeah, no problem. No problem.
Just confirming everything last minute.
Correct. Right.
Good afternoon.
Good
Hi,
afternoon,
good afternoon,
Uma.
Good afternoon,
Uma.
Uma. Okay.
Thank you for joining.
Pleasure.
मैं जानता हूँ ना सर जो है नहीं ये सर जो है मेहनती भी है और इंटेलिजेंट नहीं तो बात है मेहनत तो छोटी इनका छोटा
Please mute yourself.
मेरी तारीफ कभी कभी करता है
Dr. Shankar, please mute yourself. Good afternoon, Pavan sir. Good afternoon, Professor Srivastava.
Good afternoon.
Hello, sir.
Good afternoon, sir.
Namaskar.
Yes, sir.
पवन सर, आप आज
Yes.
मोदी जी की तरह नजर आ रहे हैं हमें। हमने सोचा मोदी जी हमारी मीटिंग में कैसे आ गए। Dr. Mary, please mute yourself.
चपन सर ने अभी ज्वाइन नहीं किया ना?
नहीं अभी नहीं ज्वाइन किया। आई थिंक
राव
वही
साहब?
डॉक्टर बाला उनके साथ शायद बात कर रहे हैं।
राव साहब भी नहीं है?
हाँ राव साहब की थोड़ी तबीयत ठीक नहीं है, ऐसा मॉर्निंग में उनका मैसेज आया था, बट उन्होंने कंफर्म किया कि वो थोड़ी देर के लिए ज़रूर ज्वाइन करेंगे, लेकिन शायद बात करने वाली सिचुएशन नहीं है, तो देखते हैं। मैंने सर को भी दोबारा रिमाइंड किया है अभी थोड़ी देर पहले, तो डॉक्टर बाला की बात हुई उनसे, तो ही सेड वी विल ज्वाइन।
गुड। एक सजेशन दूं डॉक्टर ना,
हाँ जी।
ये जैसे हमारी अटेंडेंस को इनकरेज करने के लिए हम ऐसा ना करें कि एक डे फिक्स कर दें, मतलब मंथ में जैसे लास्ट थर्सडे या लास्ट फ्राइडे, ताकि सबको पता हो कि उस दिन होना है, तो अपनी दूसरी कमिटमेंट से ये करके मतलब इधर को एक तरफ वो कमिटमेंट दे दें, क्योंकि किसी ना किसी जगह सबकी मीटिंग्स चलती रहती है। अगर हम एक वीक का एक डे फिक्स कर लें, जैसे फर्स्ट वीक थर्ड वीक जो भी है, मतलब मंडे ऑफ थर्ड वीक ऑफ एवरी मंथ ऐसे कर लें, तो देन एवरीबॉडी शुड नो बिफोरहैंड कि हमारी ये कमिटमेंट तो है ही है, बिफोर वी एक्सेप्ट अदर मीटिंग्स और शेड्यूल अवर अदर मीटिंग्स, एट लीस्ट वी कैन काइंड ऑफ कीप दिस स्लॉट फ्री, जैसे टाइम है ना। और अगर कभी कोई स्पीकर की अवेलेबिलिटी नहीं है, ऑब्वियसली वी हैव टू रिस्पेक्ट दैट कि स्पीक हो,
प्रिसाइजली मैं वही कहने वाली थी कि
प्रेफरेबली वही जैसे।
फ्लेक्सिबिलिटी उनको दी जाती है पहले
हाँ, तो जैसे
फर्स्ट।
मैं 2 सोसाइटीज़ की कराती हूँ वेबिनार जैसे, तो
जी।
हमने ऐसे ही करा हुआ है, मतलब लास्ट फ्राइडे ऑफ द मंथ इंडिया टाइम पे हमने 1:30 आफ्टरनून रखा हुआ है, और अगर हमारा स्पीकर अमेरिका से हो तो हम फिर वो 6:30 इवनिंग में कर लेते हैं। और ऐसे ही एक और इंटरनेशनल कमेटी का हम करते हैं, तो उसमें भी हमने एक डे फिक्स किया हुआ है। तो कोशिश करते हैं, स्पीकर से रिक्वेस्ट करते हैं कि ये स्लॉट मतलब हमें आप दे दो। तो थोड़ा बहुत कई बार स्पीकर्स भी एग्री कर जाते हैं, मतलब हम लोग अगर बोले तो, है ना?
बिल्कुल बिल्कुल
उससे
बिल्कुल,
अगर किसी
ट्राई
तरह
करते हैं, ट्राई
मतलब
करते
एक
हैं।
you have to try different
एक और
ways
एडवाइस
कैसे।
था करना।
हाँ हाँ, यस रवि।
हम लोग अपने वेबसाइट पे भी एक मंथली जो कैलेंडर है, जैसे आप अभी सजेस्ट कर रहे हो, उसके हिसाब से उस डे पे फलाना दिन आईएएस लेक्चर
हाँ,
करके और कौन बोल रहा है उधर ही अगर आ रहा हो, तो पूरे अगर नेक्स्ट कपल ऑफ वीक्स का हमें आइडिया होगा उसके हिसाब से, तो मे बी इट वुड बी आल्सो नाइस।
हाँ।
इट कैन
श्योर
क्लैश
रवि,
राइट मेनी
श्योर
मेनी
We
श्योर।
have to plan speakers पहले से एंड फिक्स कर
हाँ,
लें अगर हम तो 6
एटलीस्ट
मंथ्स
एक
के जो
महीने के
रेडी है तो,
स्पीकर्स तो हो जाए तो
या
ठीक है।
जी
एटलीस्ट
जी
3 मंथ्स के भी रेडी हों तो हम उस पे डाल सकते हैं वेबसाइट पे कि
जी जी
नेक्स्ट 3
जी।
मंथ्स में ये स्पीकर्स हैं और ये टाइम है।
तो
जी।
वो
तो
कैलेंडर के हिसाब से वो भी फिक्स हो जाएगा, इम्यूनोलॉजी डे भी फिक्स हो सकता है या फिर सीएमई का फिक्स हो सकता है, तो उधर ही फ्लैश हो जाएगा, तो सबको पता रहेगा कि अच्छा इस दिन ये है, इस दिन ये इवेंट है।
बिल्कुल बिल्कुल।
मतलब we have to try different ways somehow to increase the participation. At least our all EC members must commit that that day they will all be available.
Yeah, Uma and Ravi, I agree with you. Sorry, there was a phone call.
That's okay. I think next EC meeting, put this on the agenda, how all of them feel that they can ensure the participation of EC members and in their zones. I think the vice presidents also need to take some commitment that they have to encourage participation from their zone.
I think we have Professor Pichappan. Good afternoon,
Hello, sir.
Professor
Good afternoon, sir.
Pichappan. I don't know whether he's able to hear us. Sir. Sir, are you able to hear us?
I don't think so.
Yes, some issue.
He's sharing his slides, so.
Probably.
Pichappan sir, good afternoon.
Please ask Dr. Pichappan to unmute.
Okay, sir. Professor Pichappan, kindly unmute yourself, sir.
No, you have to call him, I think.
Acha.
Yeah, I think we have to give a call and then tell him.
Make a phone call and tell him to unmute
Okay, sir.
himself.
Dr. Bala, can you please do that? Dr. Bala, are you here? I think he's calling him. Both of them are on the phone. Dr. Bala, please ask sir to rejoin. Ask sir to rejoin in case he's not able to hear us or we are not able to hear him.
Sir, will be able to hear us? Or you rejoin, sir, once again? The same way. Slides are there. Slides are there. But
We are able to hear him. We are able to hear him.
Oh.
Hearing me through his phone.
Yeah, slides are there, sir. Slides are there.
Please ask him to unmute.
His voice is not clear.
No, his voice we were hearing through Bala's phone.
Oh, I see. Sorry.
Yeah.
Oh, okay.
He's still muted.
Sir? Past. Yeah, now it's coming, sir. What is sir, but you are unmute பண்ணிருங்க sir. கீழே icon இருக்கும்.
Here all our apologies for the inconvenience. I think Dr. Pichappan is able to connect with us. May take a few minutes.
வீடியோ கால் பண்றீங்களா சார்? வீடியோ கால் எடுக்கிறேன். அவர் வாட்ஸ்அப்க்கு அது வீடியோ கால் பண்ணுங்க சார் வாட்ஸ்அப்.
Dear all, we welcome you. There is some delay. Our speaker is trying to connect with us, and we will initiate the program shortly. Thank you for your patience. Good afternoon, Professor Rao. Professor Rao, can you hear us?
Unmute here.
Good afternoon, sir.
Sir, Dr. Pichappan is trying to join us. So once he joins, we will start the proceeding. Thank you.
But it's so clear.
I think Dr. Pichappan is there online. He has joined. Can Dr. Bala confirm it?
Yes, sir. It is his screen we are seeing. Let us.
Now I hear you.
Okay.
Yeah.
Great, sir. Great, sir. Great, sir. We can hear you as well. Thank you.
Okay, good.
Yeah. So shall we start the meeting, sir?
Hello.
Yeah.
பாலு வந்தாச்சு அங்க அதே கேக்குது. Hello. Taruna?
Yes, sir. Yes, sir.
Yeah. It was actually not connected inside, and then now we are connected.
Thank you so much, sir. And Dr. Bala?
Sorry for the delay.
No issues, sir. These are technical issues. These happen with all the time. Dr. Bala? He's supposed to start the meeting. Yeah, he's here.
Yes.
Thank you. Thank you, sir.
Okay. Good afternoon, everyone. It is my pleasure and privilege to invite all the senior IAS members, Professor N.K. Mehra, Professor L.M. Srivastava, Professor D.N. Rao, Professor Nibriti, Professor Bhavan Sharma, and Dr. Tharuna Madan Gupta, the President IAS, and all other senior members. So many members joined, and all the IAS members, life members, and students from across the country to this Golden Jubilee online lecture series, fifth lecture by Professor R.M. Pichapan, former professor in the Department of Immunology, Madurai Kamaraj University. So I welcome you all. To begin the meeting, now I invite Professor Ravi Kutty from Department of Biochemistry, School of Life Sciences, University of Hyderabad, to introduce Professor D.N. Rao, the senior member of Indian Immunology Society and the former president of Indian Immunology Society, the chairman of this today's function, Professor D.N. Rao. So once again, I welcome you all on behalf of the whole executive committee, and I welcome all the executive members of Indian Immunology Society who has joined this meeting and who was instrumental in making this event. I welcome you all once again. Thank you. Dr. Ravi Kutty.
Yes. Good afternoon, one and all. My name is Ravi Gutti, and it's a privilege to introduce today's chairperson, Professor D.N. Rao. And Professor D.N. Rao was the former professor and head Department of Biochemistry at AIIMS, New Delhi. And he was
Ravi, sorry for the interruption. Hemant, can you please display Professor D.N. Rao's introduction slide? Hemant?
Yes, ma'am.
Please. Sorry for this interruption. Thank you.
Can you put it in presentation mode?
Visible?
Not the complete slide.
Now check.
Yes. So good afternoon to one and all. Welcome again to this Golden Jubilee lecture series. And today it's a privilege to introduce the chairperson, Professor D.N. Rao. Professor D.N. Rao was a former professor and head Department of Biochemistry at AIIMS, New Delhi. He also served as the former president Indian Immunology Society from the year 2014 to 2018. He served also as former vice president of PIMSA. He served as emeritus scientist of both UGC and ICMR, and he had been fellow of NARMS and AP Academy of Sciences. Professor D.N. Rao's research focus had been in the area of biochemistry and immunology of infectious diseases, particularly leprosy and HIV, peptide-based immunogens, and host pathogen interactions. He has guided 30 PhD students and 8 MDs. He also guided 20 masters, MSc, and MTech biotech scholars, and his group has published more than 200 research articles in national and international journals. Professor D.N. Rao has received several prestigious awards, and some of them I'm listing here. He received Professor B.K. Aikat Oration Award of ICMR for his contributions in tropical diseases, and he received Professor V.M. Thakur Award for contribution in the field of biochemistry by Institute of Medical Technology, Surat. He received Dr. K.C. Kandhari Memorial Oration for contribution in leprosy by AIMS. St. G.S. Medical College and KEM Hospital Oration by ACBI. He received ICMR Chaturvedi Ghanshyam Das Jai Gopal Award for his contributions in immunology. Hariom Ashram Alembic Research Award for basic research in medical sciences by MCI. Professor C.R. Krishnamurthy Lecture Award by SBC. Dr. and Mrs. G.P. Talwar Oration Award by ACBI. ICMR Award for contribution in biomedical research for developing subunit vaccine in nanoparticles. He received Man of the Year by American Biographical Institute of US. Outstanding Scientist Worldwide by IBC. He received Ames Excellence Award by Nobel laureate Dr. Robert Huber. Sohail Ahmed Oration by IABS. He received Dr. Pran Nath Chuttani Oration Award by National Academy of Medical Sciences, and he received Jalma Award by ICMR in the year 2020. So with this, I request Professor D.N. Rao to preside over the session and request Dr. Taruna Madan to introduce our esteemed speaker, Professor R.M. Pichappan. Thank you.
Thank you, Ravi, and we welcome Professor D.N. Rao and thank him for accepting our invitation to chair this session. And Dr. Rao has not been keeping well, and still he has joined us. He is suffering from viral fever, so has expressed his inability to speak. So on his behalf, I would introduce Professor R.M. Pichappan to you all. Hemant, can you please show Professor Pichappan's introduction slide? Thank you, Hemant. Ladies and gentlemen, let me introduce you to our speaker, Professor R.M. Pichappan, Fellow of Indian Academy of Sciences, as well as Fellow of National Academy of Medical Sciences, Visiting Professor, School of Biological Sciences, Madurai Kamraj University, Regional Director, India, the Genographic Project at Madurai Kamraj University, Director, Research, Chettinad Academy of Research and Education, Adjunct Professor, ICMR RMRC Port Blair, and Advisor, ADNA Project by MKU and Government of Tamil Nadu Archaeology Department. His area of research has been immunogenetics and functional genomics of tuberculosis, HLA diversity in India, and first coastal migration of man through India. Trained by Nobel laureate in physiology and medicine, the discoverer of HLA, Professor Jean Dosset, Paris, France. He is a pioneer in the field of transplantation matching services in India. He started as early as 1981, offering the renal kidney transplantation matching and bone marrow tissue matching services at the MKU. Part of the project on genomic predisposition for leprosy in collaboration with Professor Adrian Hill, University of Oxford. Hemant, can you move to the next slide, please? There are so many awards Professor Pichappan has received. I'm just mentioning a few of them. Lifetime Achievement Award, Genome Medicine Consortium UK, Star Award by IBCN, Lifetime Achievement Award by Indian Association of Applied Microbiologists, Darwin of India Award by American Tamil Medical Association, Washington, H.M. Bhatia Oration Award, ICMR NIH Mumbai, Lancet's Investigator Award for Tuberculosis, Washington, D.C. Several important international collaborations that Professor Pichappan has participated in: Ancient DNA Program, Chicago, USA, Cambridge, UK, Denmark, the Genographic Project, NGS, IBM, Ted Waite Family Foundation, USA, Wellcome Trust Center for Human Genetics, Oxford, Inserm Hospital, St. Louis, Paris, Commission of European Community, MRC TV, Hammersmith Hospital, London, and several histocompatibility national and international workshops. He's guided so far 21 PhD scholars and published more than 100 impact-making papers in reputed journals. May I welcome Professor Pichappan, and I invite him to initiate his lecture. Thank you, sir.
Thank you, Tarna. Good afternoon to everybody, dear colleagues. Nowadays, I have not been attending Indian Immunology Society so often. So anyway, that is because of my drifting away from hardcore immunology to related areas, but more important for immunology as such and immunogenetics. Let me start sharing my slides. I have about 60 slides and 50 minutes. I will try to take you through my scientific journey during the past 53 years of my career. Is it clear? Do you see that?
No, sir. The slides are not visible, sir.
Then I have to again show it. Share. Shift to select multiple applications. Share.
Yes,
Is-
sir.
Okay.
Yes. Yes.
Right. Since most of you are immunologists, I will try to be quick and go fast so that we complete it in time. Maybe a minute. Okay. So, what is science? According to Albert Einstein, he called Galileo as the father of the modern science. And this, in fact, was derived from the Latin word. But however, Aristotle was the one first to practice such philosophy of science. Now, it is a systemic endeavor to build, organize the knowledge, and make predictions that are testable or testable hypotheses. Now, according to Sir Peter B. Medawar, the Nobel laureate, anybody can become a scientist, whether you are an artist or a mason or a dancer. Anybody can become a scientist. That's what in the limits of science in his book he says. That is reasonably true. Only thing is you have to be a very careful observer. 1 minute. So now, but unfortunately, science is not respected in many spheres of life. This is true even with Edward Jenner, the greatest benefactor of mankind. And when his statue was to be erected there in Westminster Abbey, the House of Lords said there is no place for commoner over there. So this is the statue of Edward Jenner that is lying there in Kensington Garden in Hyde Park, London. If you go, make it a point to see that. With nostalgic memories, I have visited there many times. Now, Edward Jenner, as you know, that is the discoverer of the vaccine for smallpox, and he vaccinated James Phipps in 1796, and the boy became immune to smallpox. He predicted the whole worldwide eradication of smallpox. And UK, in fact, legislated compulsory vaccination in 1853. And this was finally achieved in 1980. Now, the statue, the House of Lords objected statue to be placed in the Westminster Abbey. And then it was placed in Trafalgar Square and to be moved to Kensington Garden in 1862. If that is the fate of Jenner, imagine a small prize like us. Now, who is a scientist? Now, I came in for science out of sheer love for science. And if you ask me a question, do you have regrets? Absolutely not. I'm satisfied, and probably because having been reasonably successful, as you will see below. And I do have some suggestions for future to policymakers, directors of the institutes, scientific bodies, and researchers. And India requires a lot. I will do it in the end. Now, my journey through ages, I will take you through voyages through time, publications, citations versus confirmations, and I would suggest every scientist to introspect their contributions and also all the more the damages to science that each one of us have made during our lifetime. Now, India, in fact, need to become a quality-based society with a sportive, positive, competitive spirit. That is my view on India. Now, my journey started in 1971 when I joined Professor Muthukarpan as a research student. Now, this is just a time scale I have given. It is a little congested slide, but since most of you are looking at the computer screen, you can easily read that. And 1971 to 76, I was with Professor Muthukarpan. And the first column gives the various people who worked with me during various periods in different projects. Now, subject-wise, I shifted a couple of times from lizard immunology to HLA and disease, and then the tuberculosis, then the leprosy whole genome studies, then human population genetics to finally undertake the genographic project after my retirement in 2006. Now, if you take my collaborators, you know that there were a lot of nepotism in India in funding. It's extremely difficult. And the project advisory committees used to be the same people, and you will not have any chance. So I got only a couple of grants, but most of my research grants I would see on the right side and the middle are all from, as Tarna madam in the introduction said, from the Western world. Now, and I need to remember here my collaborators and mentors, and Professor Krishna Swami, and then Professor John Dosing, Professor Edwards, Ivani, John Grange, Adrian Hill of Oxford, who really produced the Oxford vaccine. And in fact, Balakrishnan worked with him on leprosy genome scan. then Philip Lagrange, Spencer Wells, and the consortium of genographic scientists. You can just leisurely, if you are interested, can go through that. I made some introspect from the Scopus, and this is the paper cited by how many people. And before that, I want to introduce to the new investigators, what are these various indices that the people use to evaluate somebody? One is called H index. It is an author level metric that measures both the productivity and citation impact of your publication. Now, for example, mine is 32 overall. So H index of 32 means that the author has published 32 articles that have been cited at least 32 times. So most nobles have only 20. Now then, the H index score between 3 to 5 is considered as good for appointing assistant professor. Score between 8 to 12 fairly standard for promotion to the tenured assistant professor, and scores 15 to 20 about right for becoming a full professor. This is as of 3rd September 2021 in the Western world. What is the status of India? I do not know. Nobody bothered about this. Okay, then there is another index called I-10 index. This is by Google Scholar. This is the number of articles published by an author that have received at least 10 citations. For example, 47 is my I-10 index, and 47 means 47 articles have received at least 10 citations. So then you look at here, since 2018, that is the next list. And this is as per every year citation they give in this Google Scholar. Now, the next one, this is much more congested. I need to explain you a bit. I have stratified all these index, citation indexes based on my funding source or collaborators. Oxford, US, Estonia, Genographic. Then Genographic, but the papers pertinent to Madurai, what we produce. The other one, Genographic Global, we are all members over there. So I will not take this as my own. Maybe our contribution might be much less, but it's a consortium paper. That is the advantage of being a consortium and may help you very much for promotions and directorships. You know that most of the directors, their name will be there in every paper coming out of that institution. Now, coming down here, Chino Madurai, Madurai 1, Madurai 2. This is, Madurai 2 is more on HLA and immunology. This one is a basic, the lizard and so on. This is the Madurai L, that is a lizard, okay? So this is the total citations that I had received. You just see it for yourself. And this 605 from Oxford is the paper on the, I will tell you in a minute, Spencer Wells, which led to the genography. And this 244 is the leprosy genome scan. Okay, then the genome also we have. But coming to the Madurai, our publication alone, that is no Westerners name in that. So in 1991 itself, we published on tuberculosis disease association. That had the highest citation, 223. I will tell you in a minute. And most of them are moderate here, and some of them are low. Okay, so paper from international collaborations yielded higher citations. Reason, maybe many. One is subjects are of larger interest, so many people look at it. Published in good journals like Nature, Science, so everybody quotes that. And many of them are multicentric. So, sorry, there is a spelling mistake. Multicentric studies. Okay, so that attracts a publication in larger and in reputed journal. Now, Madurai papers yield us less citations. Well, as I mentioned, Madurai papers cited less. data pertinent only to India, and research studies attracted much less citation since the scientific world or community is very small. Only 3-4 laboratories worked on that. Still, Madurai to TB related had a good impact, which I would say that it's a good one. Or this is what we call it as Professor Muthukarappan's lizard, garden lizard, Calotus versicolor. And he worked for quite some time or days after returning from Wisconsin. And about 12 PhD theses were written on it, believe. And I studied the thymus, I studied the thymus, its development and functions. And then I surgically thymectomized and studied the immune response. And it has only thymus and spleen, no well-organized lymph node. And it is a very good model to study infectious diseases. Then, after having become a lecturer in 1976, there was 2 options for me. One was neuroimmunoendocrine axis. I, in fact, got a placement for a postdoc fellowship, but I didn't go for that. Then another one was human immunogenetics. So I had met Professor Dose in the International Congress of Immunology, thanks to Indian Immunology Society. At that time, gave me travel bursaries, and I met him. And in another 3 months, he got the Nobel Prize, 1980, and I was there in his lab in 81, where I learned the basics of the HLA, and then having come back, offered the transplantation matching services to various hospitals, including CMC Vellore. Now, this is where the TB, the HLA association we studied, and also the twin paper, this was in tubercle, the spectrum of immune reactivity. I will just come in a minute. So HLA-DR2 association with TB and leprosy is very well known. Whereas group with the RNA-DV really showed that first on leprosy. Then HLA-DR2 predisposed for more severe PTB in sputum positive patients. And in sputum negative patients, the IL-10 studied by recall memory and its elevated response was responsible for the disease. This is what we concluded. So there are 2 pathways leading to the disease. Now, for example, in India, as far as India concerned, MTB specific interferon gamma cannot be used as a diagnostic tool in India. Unfortunately, this test was being used left and right for more than 10 years in India and to be banned only a couple of years ago. So this is actually the retrieval. Look at that. Eurasian heartland, a continental perspective of Y chromosome diversity, had 605 citations. This is Sir Walter Bodmer was the last author of that. Then major susceptibility locus in leprosy, 244. I will explain that later session. This is with Adrian Hill in Oxford. And the credit of doing that goes to Balakrishnan and Sarah Meissner. They did the whole field work as well as in the Oxford, they did the screening. Then comes the Association of Pulmonary Tuberculosis or HLA in South India, Brahmajyoti, 223 citations, no Western authors. The same way, the expansion of that using the DNA typing, 194, this is by Ravi Kumaraka. This is as of a few days ago, I retrieved, okay? So, each one of us has to retrieve and introspect what we have done. Now, as you have seen from the previous few slides, the impact making comes with undertaking research on a more common area. Now, many people really bet on only citations, but Pichappan et al. Confirmed is also a citation. I refute the finding of Pichappan et al. Is also a citation. Whereas, if we have done and identified something and published, if another group repeats exactly the same thing, That will be the testimonial for your understanding of the subject. Now, this is the second paper. We call it as a spectrum of immune response, whether it is innate or inherent or genetic was the question. Now, actually, an extension of this paper, when Brahmajyoti presented, she got the investigator award in Lancet's conference in Washington, wherein Richard Horton has written an editorial on the issue, and Brahmajyoti's photograph, everything is there in the Lancet. Now, you all know that we have a dictum called a cell-mediated immunity versus humoral immunity. This is in humans as well as in any animal system, it is there. Or you call it later days as a Th1, Th2 paradigm. Now, the clinical and the pathological spectrum are the polar forms of leprosy and tuberculosis, and especially very clearly seen in leprosy as TT versus LL. I would equate to this spectrum of immunity. Now, antigen dose response kinetics can always demonstrate such a kind of shift from one to another. Okay. Now, we ask a question whether we can demonstrate this apparent dichotomy in apparently healthy individuals. So, these we published. This is the second paper, two twin papers in tuberculosis. Spectrum of immune reactivity to mycobacterial BCG antigen in healthy hospital contact in South India. If exposure alone be the criteria for somebody to become Monto-positive, then 10% of the hospital staff nurses are Monto-negative. Why so? So what we found was in this study, it was actually on about 52 outgoing nursing students, and Monto was administered from the same vial so that there is no variation. And prior to that, we bled them, 5 ml of blood, and then tested for various antibodies, BCG, PPD, even synthetic peptides. So what we found was Monto and serum antibody levels were inversely correlated. Neither Monto nor serum antibodies can be of diagnostic value in endemic countries. Of course, in pediatric cases, a very severe Monto may mean very active force of infection. Now, so a study of 42 outgoing nursing students after 5.5 years of exposure to the highly infectious hospital environment were studied for class-specific antibodies. And this is the OD readings, and this is the induration diameter. Now, instead of individually working on, we just clustered them into 5 value Monto induration diameter here on this right side. That is how it is more even. And you just see that it is inversely proportionate. And it is much more clear with the IgG. So the spectrum, then we said it is innate or inherent, or you can call it genetic. Unless otherwise you exactly tell the mode of inheritance, you cannot call it genetics. Okay. Now, what is more important is still about 15% of them do not develop Monto-positive status in southern India, where the disease is hyperendemic. Now, having seen this, actually this thesis, Brahmajyoti's thesis, had gone to John Grant, and he had also published similar ones, and they jumped to that, and they carried out a study in Indonesia and confirmed our findings. In 92, that was in 93 it appeared. In 92, we published in 91. This Fonseca from Brazil, we read with interest the article from Pichappan et al. Regarding immunological response to antigens of tubercle bacilli in healthy hospital contexts. The authors suggested that the immune spectrum of tuberculosis proposed by Lancini et al. Could also be identified in normal healthy individuals. Their data reveal an inverse correlation between tubercular reactivity and serum antibodies. We also investigated and we confirmed the test. You just look at that picture, anti-IgG alone they did, almost looks like what we have done it. Okay? So, this paper may not have many citations, but it confirms a concept. Okay. Now, then another paper, this is from Peter Parham, actually, where we participated in that case study. Again, not all the individuals or populations are the same or similar in their genetic composition. Again, at the innate immunity level. See, this was a very heavy paper, unusual selection on the natural killer cell receptor in Africa. And 3 of our population, we have studied somewhere it is here. Okay? So, at innate immunity level also, we could really demonstrate this difference. Then the leprosy genome scan. I stumbled on Adrian Hill there at Japan conference, and that was a year that he had published on tuberculosis, and then leprosy, and we had published on tuberculosis. He used a reverse immunogenetic approach. Still, what he used that even in preparing this COVID vaccine. Now, this whole genome scan means at that time whole genome, whole sequencing was not available, but plenty of short random repeats were available, loci were available, STR loci. So once in every 250 centimargan or so, a probe was made, and about 15,000 probe or so were used to scan the whole genome. Then this resulted in, just in 2 years we had done that. And Balakrishnan and Sarah Menas did that in 3 months plus 3 months over a period of 2 months in Kumbakonam and Shaktinagar area. A major susceptibility locus for leprosy in India maps to chromosome P10, C10, P30. This was the first paper from Nature in India from Nature Genetics with a 38 citation impact factor. Now, the idea is, in a family, you have 2 siblings affected. So they are identity by descent in terms of disease. You simply ask a question for a particular locus, whether it is also identical by descent. So you saw a region in this, for example, there is a large score of 3.5. About 2 itself is considered significant. The whole range distance is associated with the inheritance. So originally about 16 different regions mapped, but we have further investigated 3 or 4 regions in depth. One problem with most of these infectious diseases are, though there is a very definite host genetic background or receptiveness or susceptibility, it is a multifactorial and polygenic inheritance. Environment also plays a major role, and other genes also plays a major role. But in spite of the fact, we could demonstrate this in one locus, and then Karitosh in 2002, again another paper we published. But you see there only a short peak, and once again, this was actually tenable only for the samples from Tamil Nadu, not from the samples from Andhra Pradesh, indicating that the genetic basis may be a difference. Now, to our interest, the ABLE and then the Canadian group, French and Canadian group, in 2003, repeated our findings in another cohort of families from Vietnam, Ho Chi Minh City, and again published in Nature Genetics and exactly confirm our finding. One difference between our Indian cohort and the Vietnamese cohort was, in India, most of the patients were TT. That means the epidemic had been going on for quite some time, and most of the epidemic is now on the disappearing stage. So rather than infectious LL, many of them tend to become TT. Whereas in Ho Chi Minh City, they did have the LL cases, and while the genes that we identified corresponds with what they have identified, in the LL, they have also identified a few more regions they have mapped. So it's very rare, and this is a very clear case that the host genetics plays a major role in infectious disease susceptibility. Okay, even if you are infected, if your immune system or immunogenetic profile is not conducive, a disease may not develop. This I will come in a minute. So even leprosy, a chronic infectious disease, has a host genetic basis. A similar way, the HIV infection does not progress to AIDS in all the individuals infected. I will come to that in a minute. Host is more important than pathogen. This is what most of the people forgot. Now, in all this, the study design is very important. A high throughput technology, a large and well-defined cohorts, a mission mode approach, not limited by techniques, money, experts, or anything, is the need of the hour. This is what I need to tell you here. When myself and Adrian Hill were there in the field, meeting all the people, at that time WHO had their leprosy eradication program. So all the household data was available, and the field workers knew which house who lives, and sometimes even married girls, they came back to their parents' house to give the sample. The question was which one you will consider as a leprosy, because there are different stages of infection. We said once there is a macroanalysis. Hello.
Sorry for the interruption, sir. Please continue.
Yeah. So, the macular anesthetic patch, if it has appeared in somebody, that means disease has established in that individual after 1 year or 2 years or whatever be the time in incubation. Then from there, they may go to TT or LL or many things, as you know. So we said, if somebody has or had now treated and relieved, but still we will consider them as a patient, because we are not going to study any functional genomics. We study only the genes predisposing for leprosy. If you have to study functional genomics or in the immunology, immunological profiles, then you need to have a untreated, uninterrupted interview into patients. So these decisions are more important in the results and what we can obtain. So now I will just tell you, this is actually somebody from Bangalore wanted, and we trained them for HLM. They offered the transplantation matching. Incidentally, they also did some HIV studies. So the data they could not really interpret, and then it came to me, and using our software, what we had developed for immunogenetics, I found that there are some issues there. So the HLA-B57 and gender, whether male or female, influence the occurrence of tuberculosis in the HIV-infected people of South India. So, in other words, if somebody is HLA-B57, even if they are infected, they do not progress that easily towards a syndrome, acquired immunodeficiency syndrome. Now, the B57 distribution in the world, if you look at B17 was called as a Telugu haplotype, a study in the Durban, using the Telugus and Tamils staying there, Hammond called them as a Telugu haplotype. Look at the frequency is about 10% somewhere here in Tamil Nadu, Andhra. Okay. So now when we did, I prepared this map in 1980 with the HLA data available as on that date. Now maybe Mera or Balakrishnan should do one for India with all the latest data. Each circle is one haplotype. Each color is a different haplotype. A1B17, A3B7, A10B8, and so on. Each circle is one group, caste group or population group. 1 to 3 means a cluster of groups. KL means colors. NR means Nada. MR means Marava. Okay? So this is the social tie. So of the Tamil Nadu population alone. So one, you compare the whole India, you just stratify and see within India, within Tamil Nadu. Look at that. You do have this A1B17 in many of the population studied. The green one only in the northern one, that is A10B8. Whereas at that time we had about 700 samples. When we stratified it into exact populations, look at that, the A1B17 appears only in certain populations, so much so the A10B8 also. In other words, if you believe that immunogenetics or the MHC molecule present the peptides and that is responsible for immune response, as have been amply demonstrated nowadays by MHC restriction phenomena and so on, then each one of them should be differentially susceptible. In other words, B17 having people, B57 people in the population, even if infected, they do not really develop the disease. Their CD4 count remains reasonably high, 400 or so. And as a result, they may live longer. As a result, they may infect more customers. Most of these people were street vendors, marginalized population, and though not organized sex workers, but they cater to the people. So this is the real trouble. Nobody looks at this kind of dynamics. In terms of evolution, the virus also develops escape mutants in this B57 individual. It is a European consortium study, and this increases the diversity of the virus also. So, in other words, the whole of the when the HIV came into India, WHO and all predicted, oh, India is lost. But it is probably the restriction of this infection only to the iris behavior group has saved the country, and it has come down the cases. Now, in terms of pharmacogenomics, these B57 and B58 is also again interesting. Abacavir, an antiretroviral drug widely used to treat AIDS, in B57 people, it develops a severe adverse reaction. Similarly, B58, its sister clade, it causes severe cutaneous adverse reaction when the gout patients were treated with allopurinol. So in the Western world, people have started using these like BIPS27 as a diagnostic test as well. So in India, still, because of the population size, these are not considered very important. So I had been waiting for quite some time, and this lotus and cactus model I proposed, this was actually, I was really prompted by the seed and the soil policy, which Dr. Rao proposed in his textbook of tuberculosis. Now, this leads to the personalized medicine as well. So, for example, you have an acute viral infection versus chronic infection. And most times, acute viral infections are handled by vaccination easily rather than chronic infection. Now, again, as I mentioned, maybe multifactorial and polygenic. and you try to control your cases and controls to the maximum, then your chances of finding the result or identifying the genes responsible for the disease is greater. Now, this is the paper that we published. Not all the infected develop the disease, a lotus and coctus model. This was in Infection Genetics and Evolution in 2015. So whatever I have mentioned earlier, I had depicted there. 99.99% of the infected do not develop this serious disease. Take it COVID or anything. Then the question is, this is what the lotus and the coctus. The lotus grows, the leaf grows in pond. It requires slushy water medium, whereas the cocti, they thrive very well in deserts and hot climatic conditions. The reason being these genomes have been selected to survive. So if it is a TB patients, HLA B57 or HLA DR2, non-DR2 patients, and a conducive lung permits the growth of the infection. So what we mean by conducive here is the immunogenome profile. For example, genetic association of ACU2 receptor polymorphism with COVID-19 spatial distribution. This has been done some Bandyopadhyay and Srivastava. So Chaubey, yeah. Now you look at the frequency of T allele shows up that case fatality rate of CFR. So whereas this is less, it is more. The fatality is more. So this kind of approach is the most required. The clinicians may not be in a position to do, but the universities and the research institutions is in a better advantage position to look at this kind of issues. So not all the infected develop the disease. So 90% infected recover without any intervention. 10% infection may be established, symptoms to varying degree. 5% appropriate protective immunity is induced among them. And less than 1% severe adverse reaction like pneumonia or cytokine storm occurs with the COVID infection. But, and then the death rate is 0.1%. But to save this one, do we vaccinate 1000 minus 1? That is 999 patients. I do not know. This is how the vaccine works. Under stress, you are compelled to use it. This is a captive circumstance. And we need to tour with the world and pay the price for it. The alternative strategy is a good population genetics approach and then personalized medicine approach. Okay, all these things, as an immunologist, I was working in the case control studies and end up with the issues of appropriate controls. I had to, in parallel, tour with the human population genetics, and migration is one of the major factors too. Now, the human migration to India, again, this was an accidental work. When I was there in Oxford, we carried out in 2001. And that was published in, this is what we did. No, Spencer Wells told me that he had tested the whole of this population, but without India, there is a blank. And already I had my samples there in Wellcome Trust for my leprosy studies and tuberculosis studies. So then with that control, within 2 weeks, we had done the test and we found some bewildering result, I will tell you. This was published in PNAS 2001. And this is the most highly cited, as I showed you in the earlier tables. So the first out of Africa migration happened some 60,000 years ago. The birthplace of mankind is Africa. And through a coastal route, a small band of people, due to some natural calamity there, they exit and then they arrived at Western Ghats through the coastal route. And finally, some of the people reached Australia. This happened 60,000 years ago. Some of the descendants of this migration we found nearby Madurai Kamraj University in Virumanda. Okay, that's the person's name. And these have been depicted very well in two of our documentaries. In 2002, Journey of Man, story told by Spencer Wells, and then 2007, story told by Michael Wood. See, this is Madurai. This is actually the west in about 26 kilometers. This is actually Virumandi. Okay? He had a mutation in his Y chromosome called M130, CM130. In the context of C haplogroup, he had a mutation. Well, this is so popular. I will explain that in a minute, the basic of that. And the project, our genographic project, we undertook after this. And it was so popular that our president, Abdul Kalam, in his online greetings to Philip Tobias in Johannesburg, he said, Genographic project launched in April 2005 aimed to carry out genetic anthropological study to map historical human migration patterns by collecting and analyzing DNA samples from 100,000 people across the 5 continents. My discussions with Pichapan was thought-provoking. So that is something very interesting. Okay. Now, this is a 2007 BBC documentary. Michael Wood, he is the person. Now he is in charge of all the British Royals palaces now. And he's simply a BSc historian who has been visiting India for the past 45 years. And his daughter's names, I think, is Meenakshi and Kamakshi. They are Britishers. And he says in this book cover, Story of India book cover, and then documentary also is there, available in web. When Marco Polo visited India in the 13th century, he described the country as the richest province in the world, a land of wonders. His observation is no less true today. Everything about India is older, bigger, more colorful, more diverse, and more intriguing than anywhere else. The world's largest democracy, a nuclear power, and a rising economic giant, India is also the world's most ancient serving civilization, with unbroken continuity stretching back into prehistory. This was in 2007. We are in 23. You know whether this is true or not now, you would have realized. Now, following this 2001, our paper in 2002, Journey of Man documentary, Spencer Wells could convince National Geographic Channel, IBM, and the Ted Wright Family Foundation to bequeath about $20 million for this public research. He called me and asked, Pichapan, can you do that for India? It was 2004, November. I thought for a day, then I said yes, because even otherwise I would have retired by 2006, June. So everything is DNA. Already we were using in plenty in our immunological studies, the DNA. So the DNA tool, using DNA tool, we studied NRY haplogroups, non-recombinant Y chromosome haplogroups, and its 17 short tandem repeat haplotypes, and also mitochondrial DNA. And I got $1 million US, and I undertook this project after my post-retirement 2006 to 2015, and that was the most rewarding because I need not be tied up with day-to-day signing the attendance register. We sampled 12,400 males from India, 220 populations, and all of them sampled by ourselves in person from all over India. All the lab, all the sequencing, about 2 lakh sequencing, everything carried out in Madurai Kamaraj University. So this is the kind of research that we require, a mode, a mission mode. No, then innovation is also the key. In our leprosy genome project, we use some cryogenic kit for getting the DNA. Each kit was about, that small tube was about $20. Now, here we said no, I said no, because we go to tribals and we sample the tribals and they should not be hurt. So simple drinking water, 1 ounce we gave, they gargled, they spit it, and we collected DNA from that. We settled that in tumblers for a minute, put it, and in the field level, we spin down, and then the pellet, we suspend it in 1.5 ml tube, put some aside, and then shipped in 500, 400 numbers to Madurai laboratory, base laboratory, wherein the DNA were extracted. When I presented this in the UNESCO meeting, one French guy came and said, please tell me how you did that. Only that's all he wanted. 30 ml water, put it in the mouth. After 1 minute, fish, and then spit it back, put some aside, bring to lab within 1 or 2 hours, spin down, get the pellet suspended in WCLB. Simplest, cheapest. Please do. So we sampled 12,040 samples totally. This is the year and country-wise, state-wise split. So why does man migrate? 1 minute I take. 60,000 years ago, a new mutation we identified. And the migration is in search of greener pasture, as do our IT personnel going to Western countries nowadays. Then finding a better niche, water bodies, safety, food availability, no predator, non-conflict areas. Once you have it, you just expand. This is what exactly happens as in the case of a tree branches. So you can imagine the Y chromosome evolution as a tree. Haplogroup, we call it. These are all simple point mutations or insertion deletion mutation. We can identify. Then it is a one-way traffic. Once it has gone here, it cannot go back. And only one copy. So predominant NRY male lineages in India, if you just look at it, this NRY as such is only one Y chromosome, and it is a unidirectional evolution, and it is responsible for maleness inherited through male lineages, and it can be used to trace male migration or ancestry. And ancestry can be traced through the branches of the tree. So you see that these three are trunk within Africa. This mutation is just the people who came just out of this Africa. F in Western Ghats of India expanded. F gave rise to H1 and L1. This is in central India. This is in southern India. And J2 and O2 and R1A, R1B, they further branched. And R1B is European, R1A is Central Asian and South Asian. O2A is Austroasiatic speaker and Tibeto-Burmese speaker. J2 is the Neolithic people. So male migration can be traced, and most of the male mediated Indian migrations have been male mediated. So that made life much more easier again. So about 220 population, as I said, each one we had an average sample of about 70. And then haplogroup, NRY haplogroup composition were compared in a principal component analysis. Look at that. This direction, these are all the Sangam literature mentioned Nilgiri tribals, Paniya, Paliyar, Pulayar, Irular, and Kadar. They have F89, which I showed you earlier, next to C. Then becomes a H. That is the one H1A. This is the Central Indian clade. And this is the Southern Indian, other Dravidian clade. Okay. So when you just look at, and these are these central portion, they are mixed up. So that shows not different regions of the India are the same in terms of their gene pool, in terms of their migration. So this is with reference to the Y chromosome, we have shown this. This is L1, supposed to be Dravidian. R1A1, this is Indo-Gangetic Dove. It is Indo-European speakers. O2A, as I mentioned, it is a Mundari, Arunachal speakers. In the process of caste formation, these migration states are mingled to varied extent. That is another confusion which you need to really take care when you study genetics or immune response. Now, not all the regional genes of caste are the same, possess varied composition of NRY and HLA haplotype. Thus, not all the genes are equally susceptible to an infection epidemic. Look at that, then we also carried out the whole genome analysis using a genochip. We developed in the genographic, and the Brahmin population, wherever they are in India, they are clustered together here as a single cluster. Then the tribals of respective area, Orissa and Arunachal, see Adi, Apathani, and so on. Then this is Borakachori and so on. Then the Tamil Nadu, Andhra, Southern Indian population, they come up here. And the Gujarati populations, you see that these are from the HAPMAP project, Gujarati samples, and with the Iyengar samples clustering them. So in other words, there is a very clear, distinct stratification of the population, and it is essentially, it is the migration and the social evolution. Then, incidentally, then I just in between 2010 and 15, I was the director of research in Chettinad Hospitals, developing research for them. And also with Andaman and Nicobar, we developed some cohorts on the Nicobaris, wherein every family had more than 2 TB patients and more than 2 unaffected TB patients. They were the ideal models to study the whole genome scan. But anyway, at that time, they did not give the money. Now they want to give the money, and Chennai TRC is working on that. Now, at this juncture, the Tamil Nadu government became more interested in the archaeology and the Madurai riverbed civilizations. And we have set up now an ancient DNA laboratory at Madurai Kamaraj University. So the purpose was the antiquity of Tamils and Dravidian. The Dravidian is one of the ancient civilizations of the world, and the Tamil has the earliest literary tradition and highest number of inscriptions available in stones, copper plates, palm leaf manuscripts, temples, and stone monuments. And Professor Kumaresan of Madurai Kamaraj University is in charge of this project. Now, the language I mentioned, so these also you need to consider in that way. See that there is a Proto-Dravidian language, it has been suggested by Atkins, originated some about 12.5 thousand years ago. Whereas all the rest of the languages, Proto-Carlinian, Proto-Uralic, Proto-Indo-European, and so on, they have all developed much later. That is the reason probably the Dravidic was entirely different in its grammar. And according to Kolipakam 2018, based on the lexical data, first hand she got all these, and they have identified all these became distinct languages some 4,500 years ago. So this further, we work on that. And the issue is one called Adichanallur, a Thamirabarani riverbed civilization near Tirunelveli, was excavated by Alexander Ria in 1903 and then subsequently by Dr. Satyamurthy in 2003. And this has been dated to about 900 BCE, that's about 3,000 years ago. So 3,000 years ago, it was a very well-established society with separate living quarters and burial sites. You just look at in the museum what all they have collected. Burial urns, megalithic burials. So this is actually Keeladi, we call it, which undermines advise all the DNA, everything. We trained all the officers for extracting the bones without contamination from the field. And then they are all being worked out now in Kumaresan's lab. Now, this is actually a ring well, terracotta ring well. So those days itself, the water harvesting technology was there. See, look at this. This is actually, they have all the gears and gloves in retrieving the bones. Otherwise, they will get contaminated with the modern DNA. So this is how deep. This is actually a burial amidst laterite. It's called a Sivakalai. So this is actually a burial offering pot. 8 to 16 numbers were inside. These are actually some rice or millets. You see, there is a rice. So based on this, the DNA of this can be extracted and then dating can be done. This is in the field. Now, a museum has also been established. Last week, it has been dedicated by our chief minister at the cost of about 18 crore rupees. It's near just about 10 kilometers away from here. So you are welcome to come and see that. It's of common interest. So finally, I have a few words to share with you. It's about 58 minutes now. The students, if you love science, do science. It is more challenging. Do not take it as a job. You will be a failure. And in India, there are only limited good places to work. So only as far as immunology or related biological fields are concerned. And then for peers, I would request HODs present or past, present all the more. Appreciate good work, encourage good work, respect quality, develop competitive spirit, thoroughly lacking in India. Indian science is highly compromised. I can give hundreds of examples of that. Egocentric nepotism on caste creed operates to the core. The most success stories come from entrepreneurs like Ella Krishna. When I visited him, when he had organized his lab, he told me, Pichappan sir, do only one thing, either become an entrepreneur or stay as an academician. Both these you cannot do. That seems to be true. It is his dedication that has taken him to that level. Now, as far as the government is concerned, as do with any bureaucratic setup, the science administration has to be thoroughly revamped in this country. Who is responsible for that? It is we, the scientists, are responsible. Each one of us have contributed equally for the damaged. Now, we need to learn from knowledge economy. This is what I learned from 3 or 4 of my international projects. It is a freedom and the mission mode, the money given to you. And about those days itself, Adrian Hill got about 10 million pounds for his work. That is how he developed all these things. Now, as far as the India industry is concerned, in India, no industry or any product is available for a quality research. Everything has to be imported. As for Indian industry is concerned, whether there is ethics, I doubt. Lot of time they are trying to only amaze the world, especially in the pharma sector. So the commoner respect the elites, but the problem is whether the elites deserve that. They don't, in my opinion. Then no retirement for science. When I retired in 2006, I never felt retired. Already I had a project. Still, I don't feel that I have retired from science. Utilize the hard-trained manpower. The country should do that for the benefit of the country. But there should be a weeding out mechanism to select only the good people. So I am remembered of a few words. Honest and sincere work will ever be rewarded. My BSc teacher, Professor Samuel Raj, used to say, "Do you still work at bench?" Professor Jean Dose used to ask me every time I meet him. Don't run for rewards and awards. Dr. Lalit Sangvi, Director, Cancer Research Institute, used to advise me. Earnestness I learned from VRM, Professor Muthukarappan. And this is the teamwork, Genographic Global Team, which really made the history. Thank you to one and all. Thank you. Thank you. 1 hour, 1 minute. Yes.
Thank you so much, sir. It was a pleasure to hear this mesmerizing talk about your journey. And I invite Dr. Archana Singh, our Joint Secretary and Associate Professor at All India Institute of Medical Sciences, to present a formal vote of thanks. Hemant, can you please display the vote of thanks slide?
Good evening, everyone. On behalf of Executive Committee of Indian Immunology Society, with immense gratitude, we express sincere and wholehearted thanks to our eminent speaker, Senior Professor R.M. Pichappan, Honorary Visiting Professor, MKU, for delivering the prestigious 5th IIS Golden Jubilee Lecture and sharing highly valuable data on the basic immunological techniques, HLA in health and disease, and your work on Y chromosome, sir. Really enjoyed it. Sir, it was really an exhaustive and interesting journey for us. We are honored by your presence and sharing of your rich experience in the field of basic immunology to the community and immunogenetics. We express our heartiest thanks to chairperson of this event, Professor D.N. Rao, former president, Indian Immunology Society, for accepting for being chairperson and nicely orchestrated this event in a scientific way. We thank all the participants across the country for their overwhelming response. Thank you all. We place on record our sincere thanks to the IT team, ICMR, NIRCH, for their seamless work in making this event a grand success. Once again, I thank you all.
Thank you, Dr. Archana. The floor is open for you all to interact with Professor Pichappan. You may please raise your hands or write in the chat box, and then we will, you can unmute one by one. We will help you with that. Yes, kindly raise your hands in case you would like to interact with Professor Pichappan. Professor Rao, would you like to say something? Okay, okay, no problems. He's not well. Dr. Pavan Sharma?
Yeah, Dr. Dr. Pichappan, good evening.
Good evening. Good evening.
This
Good.
is
I should attend some Indian Immunology Society meetings.
yeah, but the 2 days lecture, I think this is something, as Turuno already said, very mesmerizing journey to take us through right from the beginning of immunogenetics and and its application into the field of tuberculosis and, of course, the human migration. So this has been for me, it is a learning experience for me, and I thank you very much for sparing so much time from your own schedule and be with us. I was very intrigued with your lizard immunology.
Mhm.
So anything very interesting from lizard immunology which you may like to say a few words?
Yes, it has almost all the functions of as of a human.
Uh-huh.
Okay, in spite of the fact only the lymph node is not there. And unfortunately, further to infectious disease, we could not go. Somebody, you see, these kind of things should be taken up at the university level. Simply, very easily this can be done. This is how in University College London, the amphibian immunity, was studied. So we did thymectomy, almost everything. We did splenectomy, cell-mediated immunity versus humoral immunity demonstrated, skin graft rejection demonstrated, almost everything. That's what once Edwin Cooper was there, and he looked at almost every techniques at that time available. We had in our laboratory. We had used it.
Yeah.
So that gave us the strength. One thing I will tell you, the lizards, that they don't eat dead, they require live wriggling insects. So our animal house fellow used to go and collect the termite bones and put it in the cage. The problem is the dominant male, as I showed you in the figure. That fellow will eat and he will allow, if there are about 10 lizards in the cage, he will allow only his close consort to eat. All others will be starving. So we were carrying out, in fact, an experiment on starving. Then we, that to overcome this, we used to feed them with milk-made intubation tube. Then audit objected, why do you buy milk-made? Okay. Then I decided, okay, I switch on to something else. Then the Fijian immunology, cellular TRC, Chennai. And again, that started eating grains like anything, per kilo, 5 kilos of grain. Again, audit objected. Then I decided, no, human is the best one. I will go with you. That is how I did. And again, more relevance anyway.
Gives me some idea about, how to study the ecology of immunology. So, that kind,
So
that
actually,
subject.
the problem is nature, nurture. Both are equally important. That like we say in the immunology, as I said, both our genes as well as other multivarious factors. What are the multivarious factors? That's what Dr. Malavia used to tell. When should you vaccinate, BCG vaccinate a newborn baby? Yes, as soon as born, within a day, within a week, at the earliest point of time. Because otherwise, if hospitalized for childbirth, and from a staff nurse, a bug might have entered.
Yeah.
So it is the first initial infection, when MTB is commensal in India. The first bug, whether it is a virulent bug or a less virulent bug, at what stage? And this is more true with the virus also. Now you might have seen that H1N1 virus is again coming up in India. H1N1, today's paper, I think, today or yesterday's paper.
Yeah.
So they are all there in circulation. We cannot run away. Yes, COVID, yes, the panic stricken, it has happened. But in 2 weeks of epidemic itself, I wrote, it won't affect Indian gene pool that easily because it is a time-tested gene pool for 50,000 years ago onwards,
Yeah.
That is how I view that
Yeah.
in this environment.
True.
Whereas both China and Europe, it's a small subset of the gene pool, which is diversifying again.
Okay.
Yeah. Yes, Dr. Rao, please.
Yes, thank you, sir, for your wonderful talk, actually. And I want to have a clarification on the ACE2 receptor in COVID patients. Is anybody studied complete the receptor polymorphism?
I think that is one called Chaube there in, he was in Estonia. Now he is there in Banaras, Raju Raman, zoology department. That is his paper actually. Maybe, you see, the problem is like, as I showed you the genographic consortium. Okay, this I suggested a long time ago in an immunology society. You are a society. You want to work on leprosy. Who all want to work on leprosy? Have a brainstorming session. What aspect you can work? On the same cohort, all of you work. It doesn't happen in India. Each one is an individual. That is a real problem. Each one rediscover the same thing. You know, in half of my research have not been published for some reason or other. The students have gone. I cannot be doing it. We have done PEP scan in tuberculosis. We have done fluorescinated MTB survival in the macrophage. There are some glimpses of understanding, but they are not, we need to scale it up. When we want to scale it up, the project is over, you are not given further money. That's the end of the story. So there is no continuity. But as you have seen that in the Lotus and Coctus paper, my idea evolved from TB Association Day onwards, and slowly we built up.
Uma, do you want to say something? I want from Uma.
Thank you, sir. Good afternoon, sir. Thank you, Vijapan, sir. It was a wonderful scientific journey. I knew a little bit of sir's journey, but today I heard in detail, and I'm really mesmerized. Thank you, sir, for enlightening us. Sir, I also wanted to ask you that this cohort that you have built up, can this now be made available to others for some collaborative work, if we can think of something and propose to you? Am I audible? Could I
Yes.
reach?
Yes, Uma, you're very much audible. Professor Pichappan, could you hear Dr. Uma? I think we have lost one. There is some issue with the connectivity.
Yeah.
Yeah.
Maybe Dr. Bala can pass on our questions later on.
Yes.
Yeah.
Okay.
I would like to.
Floor.
Yeah, sure, Uma.
No, just because sir, as sir mentioned that, it's that we all should develop kind of a consortium kind of an activity. In
Sure.
that light, I thought that if such cohorts are available and people can work together on these cohorts, then probably we can come up with interesting data on that. So a lot of genetic, like Y chromosome sir has studied. So other genetic markers could be evaluated, and it's very hard to go to the tribal area and get these samples. So they are really a gold mine, precious samples, and if they can be, if they are available, those DNAs, then other studies can be.
Sure, Uma. What I would suggest is that if you can, write to Professor Pichappan an email about what you would like to do, I'm sure he will oblige you. Sure. And I would like to welcome Professor Dr. Hari S. Sharma, who is the founder president of Indian Academy of Biomedical Sciences. Thank you, sir, for joining us. Hari sir, are you there? Yeah. So thank you for joining us. Would you like to share something? Sir, please unmute yourself. And till Professor Hari figures out his microphone, I would like to also thank Dr. Jayanti, Dr. Himanshu Kaushal, Dr. Dimpu, Dr. Kalpana Luthra, Dr. Ratika, Pramod, Pavitra, Alagarazu, Rupesh, Saranya for joining us. Thank you, everyone. I'm sure you all enjoyed this vision from a visionary, right? And he has given us so many simple lessons to take home. And, you know, and I'm sure these very simple things are going to, yes, sir. Yes, sir. You're back.
Hear me?
Hello.
Oh, yeah, good.
Yes, yes, yes. So, Uma, continue.
Tell me. Uma Ganga? Is it Uma?
Uma, please unmute. Yeah.
Yes. So each one is retiring, you know. Balu has retired.
Uma, please unmute yourself. We cannot hear you.
Namaskar, sir, and thank you very much for this journey that you talked to us today. I had little bit information about it, but today I got to know the detail, and I'm really pleased to hear this. It's really a pleasure. Sir, as you suggested that we should do consortium activity, I was wondering if this big cohort that you have collected, can it be made available for other researchers in India to plan some research work, and with your approval, if that can be, these samples can be utilized for some further studies. Would you tell us if that's possible?
Yeah, that is the issue, you know. Actually, Genographic, when the project was conceived, no profit making, without their knowledge, nothing should be done. Recently, just last week, the National Geography has asked my other place wherein I had all the samples, that is the Chettinad in Chennai, all the sample to be destroyed.
Oh.
It should not be stored. Well, doesn't matter. Now, based on the I thought that will be useful. I requested, still I have written, but they have wound up the whole project. I do not know whether they have made this for other places also. See, all other workers, they didn't have this kind of issue because it is there in a lab, they are keeping it. Whereas I transferred it from here over to Chennai, and from Chennai it is lying there, and they want everything to be relieved. So when you ask, you please permit us to hand this over to Pichapan and all these documents, they say no, you just destroy everything. So that doesn't matter, okay? But I have very clear idea now about the Indian picture. In another 2 years, it will take. Now my first book should be available in another couple of months. That is actually, that tells you how migration resulted in caste formation. Okay, with my community as a model, and then some from the northern India also. The next one will be the whole of India, including the exact question of the Aryan and the Central Asian gene pool and so on. That will tell you the status. Now, very clearly, I am very clear and convinced about what has happened. The history of India is something different, what we people are talking. Okay? Everything has happened in India, but for very long. So the value system. I am into sociological research also. I am going to Dravidian kinship, somebody has said, but they have sampled only something. And the Dravidian Krishna married Rukmini, the sister's daughter. That is the uncle his marriage. He says, so it is Dravidian, he says. But they forgot he had not included the basal population of Tamil Nadu, which is about 40%. So I am pushing people in different directions, computer scientists, sociologists, that's how these archaeologists, Tamil people and so on, to look into. I am pretty sure, again, I am little obsessed about the kind of things that our scientists have made in this country. It is actually the damage to this country. For example, if you tell Pichapan has developed a leukemia, yes, you need to tell Pichapan. X has developed, Y has developed, no meaning in that. Okay? Now, in that consortium, what they made with the CSIR, they didn't want anybody else to be involved. They wanted otherwise everybody else to provide sample. This is another issue. Okay? Now, when Uma Ganga asked for the sample, you tell me what you want to do. I can tell you the design. What is your objective? You tell. So simply the HLA map of India, I proposed once with Rajani Rani. I went to K.S. Singh to meet ASI director K.S. Singh. At that time, he had just retired. Okay.
Yeah.
And he had Indira Gandhi's period. He had spent a lot of money, and then some 25 books have been published by Oxford University Press. When I told him this idea, he was very happy about it, and we tried. But the next person, one called Rao, was there. And in fact, he didn't want even to help us to sample anthropological survey of India. Their personnel did not want to help to sample. The same way, I had a lot of resistance from Bengal for sampling, from Pune for sampling. These are all the moral fears of saying that, oh, somebody will tell, oh, you are a latecomer, you quit India. It's not the case. See, the basis, what is the biology of the people we are trying to look at? Our purpose is not really classifying. As Uma Ganga, you asked me, you just tell me what you want to test. I will give you the design of the study, be it for HLA or for anything else. And sampling is not that difficult. I can guide you every detail we have, every people we have, and very easily just going and staying for 1 week, you can collect the sample.
Okay.
Okay?
Okay,
So
sir.
only thing is wrong, we all have failed, and then every one of us are responsible for that. As a consortium, we have not worked. ICMR works on their own, CSIR works on their own. Universities, they are orphans. And private companies, they amass wealth in another direction, like stem cell storage. So India needs a different model. India needs a different model for science, like the West. A tenure and the money to tell something.
Basically,
That is what happens in US. You never have your lifetime appointment. Yes, please tell me.
so basically, exactly the same what you suggested. I was thinking that now that we have the full next-gen sequencing platform available, if we sequence all these tribal population and other populations of India, we would know exactly the peptide binding regions and what kind of pathogens or what kind of diseases these people are predisposed to, what is the protection. So these people have not been studied. And for example, also we don't know if they need a transplantation, whether they will be able to find a donor or not. So if we have the full HLA map of these populations, it will help us understand many things, molecular basis of disease susceptibility, and also options of transplantation if they need. So the healthcare that can be provided to them could be improvised. That was the idea, that if we investigate and know their HLA map using all this sequencing.
You have a directory, bone marrow directory, right there?
No, we don't have one, sir. Here there are few private registries, but the government, we proposed to DBT, ICMR. They took 15 years to come to one common this thing. Then the Department of Health Research is discussing, but till now government has not funded. So there are private registries happening, one from South India, somebody from North, but there are few lakhs of donors. No, that does not suffice the Indian population.
Bone marrow transplants are done left and right without having good match.
So they have now, most of the centers have come up with the possibility to do haplotransplant. But still, when we have a good map of HLA, then full match transplant, the outcome is going to be long-term survival will be much better. So, but we don't know what is the profile of Indian population. So even if we want to calculate, even for kidney transplantation, sometimes we want to calculate what is the sensitization status. We can't do a calculated PRA because we don't have a population profile of HLA for all the communities of India. So my idea was that somewhere if there is a cohort available, let's begin with that. That's the approach that
I happen to review sometimes some of the proposals. Actually, you see, this is again the problem. This is the egocentric scientist problem. You see, the forensic science department is having very wide network and responsible for all crimes and everything. Now, too many funding agencies, they spoil the meal again. And somebody says, yes, no, there should be an authority, DNA authority of India. And it should be controlled only by a molecular biologist. This much restrictions. And nobody should interfere with that, should have the right to interfere with that. These kind of issues, without knowing what they are talking, these went for legislation and it was returned. And the TCGA, you know,
yes, sir.
The sequencing facility, it was a dismal failure. And it has been indicted by the Auditor General of India. I have the document. And this is, you know, commercialization of, you just give me, I will give you the sequence. So this is the same with the whole genome sequencing also. You don't need whole genome sequencing for everything.
Yeah, we don't need it.
Because the platform is there, they sell it, and then I know many institutions, very reputed institutions, purchase the array readers, clones, everything. They never use the clones. They never use the clones. They could not do it. So the company had given some 20,000 clones. Okay, then you should have automated everything automation. Then you buy that. No, the purpose is to clone all these, grow all these clones, get the insert from inside as a probe, and then use it. So the problems are only with our Indian scientists. Without knowing, understanding what they are doing, big institutions are doing it. And percentage of utility of the equipment would be less than 5%. This is what we overcame in the genographic project. I had a 384 well Tagman technology as well as 8 capillary sequencer with which we did all the work. So now, HLA map of India, yes, but the technology, I will be more keen on the technology because private companies, they use SASP now and then. They would have bought a mission and they become then slaves to that mission. They have to buy everything only from those people. That should not be done. And as far HLA sequencing is concerned, in my opinion, simple single capillary sequence is more than sufficient. Ordinary conventional sequencer is more than sufficient to do the HLA sequence.
Sir, we have more economical way now to do in the capillary sequencer. Rather than that, the next generation sequencer is more user-friendly methodology, and we get more data in more exons and introns covered, and that is more economical also, and much quicker also.
Cheaper than.
Yes, sir. Much cheaper than the capillary sequencing. Yes, sir. And
How
you
much
get more introns, exons, and we can do like 96 samples in one go. So that's really good, and we can cover 6 loci HLA, we can cover 9 loci, we can cover all 11 loci also. So it's much more economical to do and more informative because you get all intron exon information. So that's really useful to
You see, one another thing I need to tell you. Most of these were based on the Western world. We have not explored the Indian diversity.
This is why, sir, I'm asking that to explore Indian diversity, we need to do this. Sir.
Now, I do not know. I have not been tracking the latest technologies nowadays for the HLA. You just tell me the technology and the cost, then we will think about it.
So I did, sir, some tribals from Northeast, a couple of tribal populations, and also.
Uma Ganga, at this time, I want to tell you, India has committed the mistake that was because of British. You know, that is, tribals are only 8% of the population of India. For that, they developed an anthropological survey of India. The rest, 92% of the people, who will take care? Don't worry about tribal. Tribal, they live peacefully in their field. Who needs that? Bone marrow transplantation, whether a tribal is going to get it done? So follow. So you just try to concentrate on the population otherwise. Very the different languages, even in 1956, India was carved out as a linguistic state.
Yes, sir.
The premise was the people talking a particular language share a particular culture, probably particular gene pool also. Okay. So now what you do is get larger populations. And then one problem is most of the clinical studies, you don't really ask for the population. They ban it. What caste you belong to, you're supposed not to. And Indian Genome Variation Consortium, when they published the paper, no population name was mentioned. So as a result, nobody can confirm their results. Unless otherwise, you say in Iyengars of Mandiam, there is a very high risk of developing HIV because of such and such thing, or diabetes. So you take protection. Whereas a nearby population, they don't have this risk allele. They don't need to be worried about diabetes. That is a kind of predictive medicine that now you require.
So the same way, what we are getting requests from the clinician, for example, HLA B5701 is leading to hypersensitivity due to aloe vera. For that, lot of HIV patients are being sent for screening of HLA B57. But we don't know in which populations the 5701 frequency is so high. So whether do we really need to screen that population or not? Randomly, so many requests.
There are two ways of doing that. Whatever population, B57 population, how many samples you have studied now?
So we studied hundreds of them. So.
Okay, do you know the caste of their people?
Yes, maybe if I pull out the five, that will.
Oh, that is where you have to pull out that. And then look for the caste, look for the state, look for the nativity. If caste is not available, go to the village level.
That information is not available. Sir. me and sir need to have an individual one-to-one. Probably everybody else may not be interested.
Yeah,
Sorry,
yeah.
I mean,
Okay,
sir.
sorry. My computer is also shutting down off, and I don't know whether it got heated up. My battery is very poor. Yeah, please tell me.
Yeah, Dr. Hari.
Good morning to all of you from Manchester right now. I'm in UK visiting my son, so I'm talking from his home. Dr. Chipman, this was a wonderful journey which you really shared of your 53 years of experience, both in the Western scenario and taking your knowledge and the training which you have had back to India and developing an amazing center there in MKU, and tremendous contribution as far as India is concerned. I really appreciate that. I mean, I'm an outsider. I'm also about 40 years away from India now. But I do travel and visit almost every part of the country. And so many changes I have seen and observed in the Indian science scenario. So many institutions have come up, hundreds of ICERs, IITs, and AIMS, all sort of those giant amazing buildings with extraordinary infrastructure, which is even at par or even better than what we have. I work in the Erasmus Medical Center in Rotterdam, which is the #1 medical center of Europe and 13th in the world, with some 15,500 workers. What I'm just trying to point out here is that so much money spent on all of these. What is going on actually? What exactly has gone wrong, or what is still going on wrong that we are not able to, as far as India is concerned, to come up with something really worthwhile in a kind of cohort. I mean, the people do not come together, do not collaborate. They are just, pulling each other and not really. That's what I have seen it from south to west to east. What can be done? I mean, is there the role of government, or is there a kind of platform by which the people can be called upon, that the top scientists, that guys, this is for the country, which is much different in UK or in Europe. If you come to any, even Netherlands, such a small country but highly organized, tremendous, top science, we remain in the top 5 to 6 countries of the world. Any field you take it, cancer, cardiovascular, or respiratory, or why we are lacking, with so much of billions of rupees being spent, what is still not okay? I think your experience is really vital, and also a policy level. Can you give some link?
This is just the mindset of the people. And especially it is the elites who damage this country a lot. Once you have sit in a position, that's the end of the story.
Yeah, which is not the case,
And you become a CSIR chief, then for 3 years you follow one rule, and another person comes, he follows another rule, and take home baby. What is the nation required? See, under TCGA, I reviewed some project. That director came running to me. I went over there. I should have charged them at that time. I looked at all those things. I told 2 things. One, you have a subject knowledgeable person to do the job. Then that you have reference standards. Without having reference standards, they work. These are the issues. This is because essentially, as I mentioned somewhere, science has become a job now.
Which is not, yeah.
So first I said, it cannot be. Aryabhatta discovered, still we need to feel proud of it. What you have done. Follow?
Yeah, exactly.
So,
Actually, I'm
this-
from
this-
70s, 70s, 80, around 1980.
I worked in a private institution also. I thought that will be better. No, the picture is much worse.
Hmm.
I find a very gross difference between West and India. That is, in India, everything is by the government, and if at all it is a private institutions, family runs it.
Yeah.
In the West, America, for example, or even UK, university, Oxford University, Cambridge, each one is independent. Hello?
Yeah.
Yes.
Sorry,
Exactly.
my screen goes out. Something, my computer got heated up.
Yeah, we can hear you. We can hear you. Carry
Okay,
on.
so that is the problem. So Rockefeller or other things, it is by separate trust. And as I mentioned, how many institutions use I-10 or other, yardstick to evaluate somebody? You see that the people blame reservation has made this. I don't think that alone made it. The quality compromise at every level.
Yeah.
That's what I feel.
Yeah.
I belong to a business community. I know to calculate.
No, I was just wondering
I
how
will tell you.
Yeah.
Having seen the nepotism and the way in which the science functions in India, though my children have studied science and everything, I never wanted them to come into white collar. They are all business entrepreneurs. No, because it is not worth it in this country.
So, I mean, how to enhance the collaboration? Because I think, in my view, like in the West, people may not be really friend or sitting in the evening drinking beer together, but professionally they are, they are together. Surgeons, clinicians, and top scientists, they are together professionally. And
So
how
yes,
to enhance that?
that is
Which
the
is
difference between the developed society and developing society. See, it depends on the leader of the team. See, Vice Chancellor in Tamil Nadu alone requires 28 Vice Chancellors. All compromised. And the political parties, they administer. So it has to be made independent, but the problem is many of the stalwarts, they all got some money, 1 or 2 crores from the government, and started one institute in their name.
Yeah.
Subject.
Yeah, exactly.
So that, and then no good people available, then they simply appoint somebody, and then a question of survival, publish some papers that need to change. See, my son has the terminal, shared terminal. Hello?
Yes, sir.
Yeah.
Yes.
Yeah, shared terminal. So they have to qualify themselves periodically.
Yeah.
You know how to do it. I told the people, but they don't. They hear that. This should be applied everywhere. And they say in the SEBI, okay, you should qualify this test, otherwise you cannot run the outlet.
Yeah.
Then these people study, and then some 3,000 rupees they pay at the particular date and particular time, particular appointed hour. You go to a particular place, sit there, half an hour ago, log in, then they open up, then the questionnaire opens, or by 2 o'clock means 2 to 4 o'clock, you answer everything. At the end of that you have completed, or 4 hours is over, you click yes, and it gives you the score, how much you have scored, whether you pass or not. Next minute, it gives you a printout certificate and comes up. Unless otherwise this is done in education, education will not improve. And getting a foreign university into India is not the solution. And take from me, they will never set up their, shop here because their income will be lost. First question. So no, they will not come here. They will take everybody from here over there so that they really live.
Yeah.
Education has become a saleable commodity.
Brain drain.
Yeah. No, well, I won't say brain drain. They otherwise America would have closed down all their colleges a long time ago. The same is true with London, UK. I once discussed the London School of Hygiene.
Yeah.
Can we have a parallel course there in Madurai Kamaraj University? We have all the facilities. Faculty from here can go over there and we can train the people from developing countries. It will be much more cheaper. Nobody wanted. Unless otherwise you get that diploma in London School of Hygiene, you are not eligible for promotion in WHO. So nobody gives anything out of love for you.
Right.
There is only the scientist has to join together and do it.
Yeah. I think the responsibility actually is with the scientists.
Yes.
In making a kind of, I think the consortium, you may call it, or coming together, subject-wise or expertise-wise.
Government, for example, you know how many people we have for funding a project? Everybody does the same thing. Indian Council of Medical Research, Department of Biotechnology, CSIR, Council for Scientific and Industrial Research. Not even DNTP or tag DNA polymerase is available in India.
Oh my God.
And we promoted one company called Tarsen earlier days. Very quality compromise, but he will also sell for the same price as the Corningware. That is another issue.
If the mindset of people changes, if we all decide we will work together and help each other, then the government will be all right. But unless our Indian community changes our mindset, if we don't help ourselves, nobody will help us.
That's what I'm actually.
No, the thing is, ICMR, then DBT. DBT says everything is, I will do it. Then CSIR, DST, everybody fund for, why don't you have single platform and invite proposals for tuberculosis research and then fix up the priority. Work on the priority basis. ICMR has a TRC center there in NIRT, there in ICMR has 110 institutes. Each one 110 directors, each one for one disease.
Exactly.
Each one has elaborate lecture halls, rooms, guest rooms, everything. You follow?
Yeah, exactly.
So that is the kind of problem. And it will not be available for anybody else. It is on the whims and fancies of the director concerned.
So may I
think we
interrupt this session because we had booked it for 2 hours, the platform, and we need to close it down. And sorry for this interruption. It was a very interesting dialogue going on with Dr. Pichappan and everybody. And we thank you once again, sir. From the bottom of our heart, you have enlightened us, motivated us, and we wish that we all can imbibe some of the values that and the guidelines that you give us, and hopefully we'll make a change. Thank you, sir.
Okay, thank you, one and all.
Thank you.
Thank you. It was so much lovely to see you all. Kalpana, Dr. Rao, Taruna, it was wonderful to see you.
Okay.
And a wonderful talk. Thank you, Dr. Karna.
Thank you, Dr. Kalpana.
Thank you.
Okay.
Bye-bye.
Okay, bye-bye. Hemant, we can close now. Thank you.
Thank you, sir. Thank you, Tarna. Yeah.
Thank you all.